Yeah, it's kind of intimidating here because people are way too nice, and so I can't possibly live up to all this, but I'll try my best. So here we go. Let's see what happens. So that's the you heard the Thailand. I should have realized that about the red queen. The red queen is an expression that occurs in evolutionary biology as well, by the way, as well as universities competing for funds. That you have to run as fast as you can in place, just stay in place. So I'm an experimentalist. I'm not a theorist. Despite I coming from Princeton, where everybody thinks we're all Albert Einstein. That's not true. And so there's a lot of really great students I had over the years, and as long as somebody came up with Jim Sturm, for instance, some of the students here that you see here actually are students of Jim Sturm. I work with electrical engineers a lot in this area. And I've also spent some time here at Hong Kong University of Science and Technology. I'm a postdoc here. So anyway, it's a big group of a bunch of people that have been involved in this over the years. So this is supposed to be a general lecture, so stop me if I don't know what I'm talking about. If you don't know what I'm talking about or you don't know what I'm talking about, let me know. I'll be trying to clarify things up. We're going to be talking about DNA, which is sort of the element of heredity, and it contains the code for you, as you probably know. It has about 3 billion base pairs in the human genome. Every one of your cells has about 1 meter of DNA, which is enough DNA when stretched out to reach from the center of the sun to Pluto on all your cells. It's that much stuff. So that's the code for you in principle, and there's the base pairs you see right here that match up AT and GC. And this is a scale model of the one of the human chromosomes. This is this guy in Darwin, Minnesota, appropriately enough, and he made a world's largest ball of twine. It turns out it's about a scale model of what the human genome actually would look like if we rolled it up using twine. And here's a chromosome here with the DNA exploded around it, and here is a single place inside that thing. So what's happened over the years is we've learned to sequence the human genome. We now know more or less all 3 billion base pairs on the human genome. And what's going on here in the human genome is that 3 base pairs form a triplet code. There's it right there. And 3 of those base pairs then code for a particular amino acid. So the DNA has a code in it, which consists of 3 base pairs, a triplet codon, and then a particular combination of those things gives rise to an amino acid, and that forms a protein. And then those proteins do the work. And I think I want to show here a movie. Let's see if it runs here for us. This is a particular molecule we're going to look at here. This is like sort of this is actually an antibiotic you're looking at here called Cipro, ciprofloxacin. Here's what DNA looks like right here. This is the antibiotic, which is binding to the DNA. That's how it stops the cell from functioning. This is the protein within the complex. This particular protein binds DNA. And these little red things right here represent evolution. There are amino acids that have been changed by a mutation. So you can change that sequence of 3 codons, you make a different amino acid, and then that influences the way this antibiotic binds the DNA. So it's a very, very complicated thing going on here in your body. It's really quite remarkable. So a mutation means that there's been a change in the base pair sequence that codes for a protein or a binding site or something like that. And that changes the behavior of that particular thing. Now these mutations that happen, they happen all the time. You're being mutated right now because of cosmic rays coming down through the air. You're being mutated because of some of the food you ate. If you're a smoker, you're being mutated right now in your lungs. These mutations happen all the time. And the system tries to work with that. So they can take many forms. Now I want to let you know that I'm going to be talking about simple things, just replacing a single base pair with another one. That's called a SNP, a single nucleotide polymorphism. It's kind of a complicated word, that's a SNP. But you should not be misled in thinking that SNPs are everything. There's all sorts of genomic rearrangements that can happen in your cells. So we're going to look at this one particular thing because I'm talking to a general audience, and I'm going to try to make things simple. But you don't be misled by the simplicity, okay? But the story, nonetheless, will be sort of okay, but I'm trying to deliver it. So last night I was having dinner with, Shirley's Puching, is her middle school she went through in Hong Kong. And I was asked a question when one of the people at the middle school in her classroom said, wait a minute, shouldn't the title of your talk be how do organisms develop resistance to drugs, a physics perspective? Why did you say why, right? That was a great question, actually. Why did I use the word why instead of how? So I pretended I didn't understand his English and I escaped it. But it's a good question because the why question is sort of forbidden in physics, right? Physicists aren't allowed to say, why does a proton exist? It just does, right? It's there. But in biology, it's a legitimate question. Why do these things happen, as well as how? So I'm going to try to explain that a little bit as we go along here.